Hi, Professor.
Hello.
Professor, I have a question about the home lab.
Mm-hmm.
I just figured that I needed 3 days to do on my agar plate.
Yeah, so a number of people are saying that they won't be able to make it at that time. That's okay. Well, we got an exam coming up. I'd rather you focus in on the exam.
Oh, okay. Thank you so
But
much.
I'll move back the deadline on that.
Okay, thank you.
All right, it's 2 o'clock. I'm recording this session in case you guys want to know. It is September 20th, 2020. This is the second out of 2 study sessions that I'm holding. Hello, Daniel. Michael, Katrina, got a number of people, Ryan. Oh, wow. So we got about 9 people on. Does anyone have any specific questions? Okay. Sharing my screen a little bit. You may or may not see my screen. Okay. So guys, I'm trying to put it in the chat, but we have a review document that hopefully you guys have all seen. It is not going. All right, so what I put in the chat is the review document for the exam. The exam itself is going to be roughly about 30 multiple choice questions, probably about 6 kind of short answer questions, and then 1 kind of longer essay question. You can find examples of the multiple choice questions if quiz number 6 basically looks at our database of questions, and what I did is I took some questions out of the database and I modified them, so they're not going to be exactly the same, but they probably will be similar. For the short answers, I kind of went through and I picked topics that hopefully weren't covered by the multiple choice, and we'll put those up there. One of the short answers is going to be generating a figure on your own. If you want me to show you how to do that, I can do that for you. And then one of the last part of the exam is going to be the essay question. And so I'm budgeting for all of you guys. Hello? So I'm budgeting for all of you guys is roughly 30 minutes per section. The exam itself will start at 11 o'clock on Monday, or 11:10. And once you start, you got to 9 minutes. Or 11:10. You got to 9 minutes. Yeah. Okay. All right. So does anyone have any specific questions?
Hi, Doc.
Hi, Andre. How's it going?
Good. Can you go over like what taxonomists have trouble, like why they have some difficulties with classifying bacteria again? Is it
well, the big thing when you when you classify things, you kind of look at their differences. And so back in the, an early age of taxonomy, you're looking at, you can tell a difference between a rabbit and a snake, right? One's a mammal, one's a reptile, et cetera. And so you're looking at these kind of gross morphological characteristics. What are the 3 possible shapes for bacteria?
Let's see. Like for bacteria,
Yeah.
like cocci, rods, and cocci, rods, and
Spiral.
And
spiral.
spiral.
Rockets,
Right.
rockets.
Spiral.
So again, when you're looking at microorganisms, there's really not, again, at least in the early age, there wasn't a lot to tell the difference between these guys. And so that's one of the reasons why, again, the 3 domain system didn't come up until about the late 70s, early 80s. And then that's where they started being able to tell the difference between them. Does that make sense?
It does, yeah. So basically, everything before before they classified them into 3 domains, it was pretty much difficult because of their different shapes.
We didn't have the technology to tell the difference, right?
Okay. Yeah.
So you saw bacteria, and there's rods, cocci, bacilli. You had the gram stain has been around for about 100 years. And so that's one of the big reasons why they kind of use that. But there wasn't, so why it was hard for taxonomists for a long time to differentiate these things is just because we didn't have the tools.
Okay.
And now that we can sequence and we can look at proteins, we can look at DNA. There's a whole bunch of different ways that we can identify these things. And that's one of the reasons why we have a lot of this out.
And was that part of the reason why you said Bergey's manual, it's good, but it's not good?
Yeah, so the Bergey's manual is basically, it's a lot more for like clinicians.
Okay.
As if, when you're a clinician, you really want to be able to identify something, right? Because you want to be able to kill it. And sometimes you don't even care. As long as you know you have an antibiotic that will kill everything, then you're going to use that antibiotic. You're not that interested in knowing what it is. But is there a situation where it may be more important to know the relationships of these guys? And that comes in especially when you have like a new disease and you want to treat the disease.
Okay.
Yeah. So that reminded me of something. Yeah, but that's about it. That's about what you can see with that. Okay. Does anyone else have any other questions?
Yes, Professor. Can you kind of like differentiate in a very simple way the acid fast stain and the gram stain?
Okay. So just kind of real quickly, the gram stain is what you're doing is you're trying to, in the gram stain, you have a substance called the peptidoglycan.
Mm-hmm.
And your gram positives are going to have a very large amount of peptidoglycan, right? So when you're doing the gram stain, you stain it with crystal violet, then you put the mordant in there. And what happens is those two bind together and it's bigger. So it's harder to come out of the peptidoglycan. So you do the wash, but the gram negatives that have very little peptidoglycan will kind of wash out. The acid fast, what we're looking at is lipids. So there's some bacteria have a very waxy outer coat, and it's hard to get the crystal violet inside of them, and you really can't tell if they're gram positive or gram negative. And so you use the acid fast bacteria. Hello, hello.
Teacher. That's my niece.
Okay.
Cool.
So to determine the doc, do you need to apply them both? What if the gram stain does not?
So what I would do is if you're looking at something and the gram stain doesn't look like it works, you're having a real hard time gram staining it, I would do the acid fast stain.
Okay.
But it's, for most part, most things will be either gram positive or gram negative.
Okay. Thank you.
So the mycobacteriums and stuff like that, tuberculosis is one that doesn't stain with the gram stain. So that's why I have to use the acid fast stain. So there are different types of differential stains.
Okay.
Okay. Anyone else? That was a good question. Thank you, Amy.
I have a question.
Yeah.
I was hoping that you can clarify the quorum sensing in biofilms. I understand the purpose of biofilms and how it gives advantages to like pathogens and disease and everything, but I can't wrap my mind around the quorum sensing part. There's a disconnect there
Okay.
for me.
So where the quorum sensing comes in, well, do you know what a quorum is? You know, we use it a lot. What basically when you have a quorum, you have enough people to start a meeting. And that's why they look at getting quorum, right? So like the United States Senate, if you only had 10 people in the room and they voted on something and made it a law, right, that wouldn't necessarily be fair. So what they have these rules are, you have to have a quorum, you have to have a certain number of your total body in order for that body to work. And so what's going on with quorum sensing is kind of the same thing. The bacteria, before, you know, they can't, if there's too few of them, they can't make a biofilm. And so what they do is they kind of wait till there's enough of them there, and then they get together and they make a biofilm. So what they have to do is they have to have a way of counting or a way of kind of taking roll of kind of who's in the area. And so what a lot of times bacteria will do is they'll produce a specific molecule, and when that molecule reaches a certain density, then the bacteria could produce a biofilm or produce a fruiting body or something else.
So if I understand that correctly, it would be like a threshold. There needs to be a threshold that needs to be met in order for it to work, or in order for an action to be made, meaning the biofilm to generate
Yes.
or start working.
Yes.
Okay. All right. That makes
Right.
sense then. Thank you.
That's good. Oh, no problem. Those are real good words. That's a real good analogy or a synonym that you used for it. Yes. Anyone else?
Yes. Or does anyone else just have any questions before I ask something?
Looks like you're up for it.
I just want to go over, maybe if you can go over some more examples of math problems, like more maybe like the percentages again. I want because I don't think you graded it yet, so I want to see if if my answers were correct.
Yeah.
Like for example, the one where it says iodine is used as a mordant in the gram stain. It was on the Google Docs you sent us.
Uh-huh.
Yeah, and then 6 grams of potassium iodide to 200 milliliters of water. So I'll
Okay.
go over.
So what percent is that? So I'll put it in the chat. So you
Okay.
have 6 grams of iodine per 200 mils.
Uh-huh.
The density of water is basically 1 gram per mil. So roughly 200 mils is equal to 200 grams.
Okay.
Okay. So sorry, that's some stuff that you got to remember from chemistry or physics and stuff like that you should know. And so what you want to do is what percent iodine is it when we add 6 grams to 200 mils?
So would I do 6 grams divided by 200 milliliters and then times it by 100? I got 3%.
Yep.
Oh, okay.
Yeah.
Okay.
Great. Thank you.
Oh, okay. Good.
Yeah. So that kind of goes back and forth. I mean, to me, that's percent. It's like doing a tax problem, right? Or maybe not. Yeah.
What percent of people, or I don't know.
So, but in that one there, we're kind of looking at solutions. We may be looking at percentage in a population. Okay. The other thing that I do want you to be able to figure out, so if we have something that's 200 meters. So, yeah, so 200 millimeters equals how many microliters?
Yeah. Micro.
Not microliters, micrometers.
So that means I'll move the decimal twice back. It's 1,000.
So what's bigger, milliliters or micro? What's bigger, millimeters or micrometers?
Millimeters.
Millimeter.
Millimeters.
Milliliter.
How much bigger are millimeters than micrometers?
Isn't it three decimals?
Yeah. So,
So it's
but what is?
200,000?
No,
No.
1,000.
Oh.
Right. So you got the tens, hundreds, thousands. So 200 millimeters is equal to how many micrometers? And so what you would do is you would move that decimal place over 3.
Yes, 200,000 micrometer.
And that would be 200. Yeah, 200,000. I put up in the chat. So I will probably ask you some type of conversion problem like that. And then sometimes I may have you like do it in lengths of E. coli. So I give you another thing to kind of look at and stuff. But for the most part, because it's a micro class, you do need to know what micro and milli and in meters are, you should do it in grams as well.
Dr. Cap,
Yes, Ryan.
I thought for when you're adding the iodine to the water, do you want to include the extra 6 grams of iodine that you're putting in?
Yeah, typically you do that as well. I wasn't going to get into that because that adds another level of complexity, and I figured you guys would get through that in your chemistry class. But typically when you make up this solution, what you would do is you would use about half the volume. So you'd put about 100 mils of water in a vessel. You would add your 6 grams of iodine, and then you would go up to 200 mils.
Okay. I just wanted some clarification on that.
Yeah.
I just wanted to make sure I was doing the math right.
But again, so you're right. It's in reality, it's going to be somewhere a little bit less than a 6%. Than 3%, but not that much. And so especially if I'm doing multiple choice and stuff like that, you're not going to see that.
Okay.
But very, very good.
And can you clarify, like identifying the dilution on a serial dilution? Because are you asking for the ratios, like the 1 to 10, 1 to 100, or is it?
Yeah. So if I do a series of 3 dilutions, right? A 1 to 10. So this is going to be in the chat. Another 1 to 10, and then another 1 to 10. So and each of those steps, that's a 1 to 10 dilution. What's the total dilution?
For the final, so it'd be 1 to 100,000?
Where'd you get 100,000?
I was just looking at the chart for the serial dilution charts from your lecture.
Okay. Now, well, I'm giving you in the chat, I gave you a little bit of numbers. So I do a 1 to 10, a 1 to 10, and a 1 to 10 dilution. What's the total dilution there?
1,000th.
1,000? Yeah.
1,000.
Yes.
So you
that's
wouldn't
1,000.
count the original culture then, right?
Well, so this is just a dilution, right?
Okay.
And so if your original culture, so if the original culture had 300,000 bacteria per mil in it, okay, how many bacteria per mil do you have after the serial dilution?
And then that would be 300 after 3 dilutions because you dropped the 3 zeros.
Yes.
Okay.
But I could also ask things. So if you have 300,000, how many bacteria per mil do you have after a 1 to 10 dilution?
You would just divide it by 1,000 then, right?
No, because you're doing a 1 to 10. You got to figure out what you're. So after the first dilution, right?
5?
It would be how many?
Is it 5?
30,000?
30,000
Dropping 1
bacteria.
0.
Yes.
Oh, yeah, I see.
Let me get the comma. Oh, my comma's not working. Okay. How about after the second dilution?
3,000?
And then after the third and final dilution?
300?
Ah, okay.
Okay, so do you get it? There's a pattern there.
Yeah. Wow,
And
thanks.
so a lot of times you could kind of follow that pattern and see what's going on with it. Why do we do serial dilutions?
Because like sometimes the original culture in the media can be too many to count.
Yeah, that's true. And it's a way to count. It's a viable count.
Yeah, so like when you're doing the Coulter square, it's a lot easier to count like 3 bacteria after a few dilutions than like 300.
Yes, but then you gotta look at the statistics, right? So when you're doing a dilution like that, how many bacteria do you want to count?
30, between 30 to 300?
Yeah, roughly 30 to 300. I've seen 25 to 250. Those numbers are all similar. I would take those numbers, right? Because you're at that point you're kind of doing statistics. So if you have 1 out of 2 dentists, recommends whatever, some dental paste, some toothpaste, right? If you ask 2 dentists, you have 1 out of 2, but you don't, that number is statistically insignificant, right? So you have to ask somewhere like 100 dentists what they want, and then you can do those numbers. And those are much better numbers.
And I guess that's why it's important also, why it needs to be, the bacterial culture need to be fresh, like no more than 2 days, because it can affect the dilutions, growth.
Well, what's going to happen to your culture if you're leaving the test tube for a couple of days?
Probably just die, right?
Yeah, it starts to die off. You have that growth phase. They'll grow exponential as long as they can, and then they hang out in the kind of a stasis period, and then after a certain while, they'll start to die out. It depends on the species and stuff like that, but a lot of the pathogens that we see in human beings and stuff like that, they'll die relatively fast.
That was that log phase, huh?
Oh, so the bacterial growth, right? So what are the phases of bacterial growth?
Let's see. I have it in my study guide.
And then, does anyone have a figure and they could share it with us?
I could Google it really quick.
Yeah, Google it.
There's
really quick.
it's
Lag,
like the
log.
sedentary stage,
Log. Sedentary.
then log, and then I know it plateaus after that.
Stationary.
Yeah, there's several names for it, and we'll get to one. Was somebody going to Google that and share it?
Someone put it in chat.
Oh, they put it in chat. Let me see.
So if you were to ask like a short answer about that and we don't get the precise name of the stage right, but we still describe it correctly. I mean, I don't know if that's a question, but if
Yeah.
it were, would that be acceptable?
So yes. So what I would do for the short, but trouble is multiple choice. Each of them is going to be worth 1 point, and you either get it or you don't. For the short answer, what I would do is, I can assign each, trouble is the short answers. I think they're going to be worth like 5 points apiece. However, if you forget the word That's not the best thing. You forget the word, but you describe it or give me a picture and, and do everything else, but give me the word, then I can give you some partial credit on that.
Okay.
Oh, I'm not getting a screenshot from this down. But let me do, so let me, I'm just looking up bacterial growth curve phases. Do you guys see this? Do you guys see my screen?
1 second. Let's see.
Yep.
Yeah.
Yes.
Oh, sorry. Well, all right. So the figure from your book and the main one that I want is this one right here. Let me see if I can make that a little bit bigger. And I'll move it over. So you have something that's called lag, so when you first start out, and so you're looking at the logarithmic of the cells over time. And then, so what happens in the very first beginning, when you first do your inoculation, the bacteria really don't grow all that much. Does anyone know why?
Is it because they're still trying to get used to the environment?
They need to like build cell walls and stuff, sort of get healthy before they start reproducing or whatever.
It's preparing itself for growth and division. It's ramping up its metabolism.
I'm liking that one a little bit more. So we're kind of doing specific, we're doing specifics. So what happens is that it depends on where the cells came from, right? So if the cells came from, they were starting to die out and stuff like that, they need to get a little bit healthy. They're taking in some nutrients. The nutrients may also be different than the ones that they're used to. So maybe they were growing on lactose before and now they're growing in glucose. They have to change their metabolism so that they can take advantage of that new material. Once they do, they will start growing exponentially, or what we call logarithmically. Can you tell me what that is? What's growing what's growing exponentially?
The bacterial cells are rapidly dividing and growing in numbers, so there's more of them.
Yeah, so give me what that is. So
Binary.
that's true, but that's not the best answer.
Binary fission?
That's true, but that's not the best answer.
They're doubling every generation time.
So they're doubling in a generation time. And so your generation time would be n to the second power, right? And so if you started out with 2 bacteria and their generation time is they double once every hour, how many bacteria would you have after 3 hours?
At least 800.
No.
Sorry, can you repeat the question?
Wait, how much
So
are we starting off with?
You're starting out with 2 bacteria. Well, let's make it easy. 1 bacteria.
Okay.
You have a doubling every hour. So let's make it easy. After 3 hours, how many bacteria do you have?
8.
Okay. But let's go back. After 1 hour, how many do you have?
2.
2. After 2 hours, how many do you have?
4.
4. After 3 hours, how many do you have?
16.
8. Okay.
Yeah, sorry.
So each time, hopefully each cell is dividing up into 2. So more than likely, I will give you some type of generational problem like that. But again, I probably won't give you a really hard mathematical one. I'll try not to do that.
So like the one on the activity we did when you asked us how long it'll take for the bacteria to double, or how long it would take for it to go to 1 million.
Yes.
So I put for the answer is in between 260 to 280 minutes because the range is like 819. And then
Yeah, no, that's not enough.
Yeah, so like
200
60
minutes, that's, yeah. So it usually takes, it takes somewhere around 20 hours or so.
20 hours for
Yes.
1 million?
To go to 1 million, to go from 100 cells to 1 million.
with the every 20-minute doubling rate?
Yes.
Oh, what? Really? I think I did it wrong then. Okay.
You can math it out if you
What was
remember
your
the
answer,
logarithm
Andre?
formula, but
Well, so so the number of cells I went from 100, and then I did every 20 minutes, so 20, 40, 60, 80, 100, all the way up to 260. I already got up to 819, 200 cells.
Wait, what was your time?
260 minutes? Or did I not double my minutes correctly?
I don't think you did the math right there because that's
What
like
problem are you talking about?
5 hours almost.
The one that was in the math worksheet?
Yeah.
Yes.
Oh, I got closer. I think I got 12 hours.
I got like 14 or something.
14,
Yeah.
I got like 13 and a half
40
or
or
something.
1420 and 1440 between those two.
Yeah. 'Cause
Okay.
So that's more than 200 minutes.
Okay.
But again, on all those things, what you want to do is, if you're doing it, put in your equations what you did, right? Because if you just say the answer and it's wrong, then it's wrong. But if you said, oh well, there's you have a million, if you put in what you're working on, and I can see it, I can give partial credit.
Yeah, you want to see can you see what I did wrong? Can I share real quick?
Sure.
Here, here. This is what I did. Maybe I messed up on the time.
Are you sharing? You should be able to share. I'm not seeing
Yeah,
what
I am sharing now.
I'm not seeing it.
Let's see.
Now try and share.
Okay, let me try. Let me do it again. Let's see. Stop sharing, and I'll share it again.
So in this one here, you made a table.
Yeah, on Google Docs. Because the question said, oh wait. Every 20 minutes, doubling time. Yeah, okay. 3 per hour? Oh, maybe I did that. I don't know.
Yeah, I don't know what the numbers are here. So after it gets like that, go over and look at it again. I'm not a math person. Don't care that much, but it looks like so after your first hour, it looks like your numbers are right. So you went from 100 to 200 to 400.
Yeah.
To 800 to 1600.
Oh no, Andre, you're right. You're right.
Yeah.
Because it's between the 13th and 14th generation. So it's between 4 hours and 20 minutes and 4 hours and 40 minutes.
That was right?
Yeah.
Oh,
We did the math on it and we got 265.77 minutes. So it's
good. All right. Okay, good.
You are right. Sorry, my
we
bad.
did the math and we got that too.
Yeah, I guess we're thinking of generations, not the hours.
Oh, okay. That's what I was thinking. I was like, wow, 13, 12 hours? Man, I'm way off.
And actually, I usually do this. I usually talk about 1 billion cells per mil. So this is a little bit, and I usually start out a little bit more. Depends on when you start out.
Okay, cool. Thank you.
Yeah. So you look at the generations, not the hours. So
Okay, cool.
no, sorry about that. But it's a time test, and rather, and when people do math, things really slow down. So again, when I'm doing this on the, I gave you the chart because I gave you a lot more time and stuff like that to do that kind of materials.
Let's see if I have any more math problems. Anyone else have any questions?
I actually had a question.
Okay.
So, going back to that practice for exam, the one with the short answers,
Uh-huh.
I think there was like give a diagram of a prokaryote, point out the cell wall and the the plasma membrane, but then you also have to show whether or not it's gram positive, and there would
So I
have to
think
be
that
like
one was a kind of an either or one.
because like
Let me pop that one up.
I was using this as a diagram.
All right. Well,
Unlabeled.
why don't you share? Well, you need to share your screen.
Hold on. I think I have to find a better image.
So what I was doing with that particular one is I wanted you to be able to, what I want you to be able to do is to generate generate an image and label that image for me. And so
Okay.
what I think
So it
I
has to be
wanted you
drawn.
to do is to tell me whether, so generate a picture of a prokaryote and then label and show the, label and show the plasma membrane and the cell wall.
Okay. I just
And then,
wanted to make sure that
you know, again, there's also, you know, if you're doing that, you could also tell me whether it's looking at the cell wall structure, you can tell whether it's gram positive or gram negative.
okay. So
But what I
just gotta
want
make
you to
sure
do
that's
is to be able to give me just
diagram.
that figure. So if you just get something from online that has 10 things labeled, I'm going to take some points on it because what I want just labeled is what I asked for.
Yeah, I would use an unlabeled structure. That was just a close model of what I used. I can't find the image right now, but okay. So I just need to make sure that there's enough detail in the cell wall as well that I can tell that it's gram positive.
Yeah.
The layers of the peptidoglycan.
Yeah.
Okay.
So, you know, again, when I'm looking on all these things, what you really got to do is you got to think about what is he asking? And you have to only answer what I am asking from you. And, you know, and a lot of times, sometimes, you know, having extra information is not, you know, it's not necessarily a bad thing, but when it's confusing what you need to know, then I'd rather kind of not see that. Also realize that if you put stuff in that's not what I'm asking for, but it's wrong, I have to take points off it, right? So it's kind of, you know, it's kind of like a little bit of a lawyer thing. If you're not really sure, don't put it in. Because yes, does it look impressive when you give me this big long answer of stuff that I'm not asking for? Yes, it does look impressive. But if it's wrong, I have to take points off of it. Anyone else?
I think Andre's got another one.
Yeah, I got a lot of them.
Yeah. Let's see. Just want to make sure my.
Well, Katrina, what'd you come for? What are you looking at? You just wanted to listen? We're not hearing you. Oh, your mic isn't working. Okay. Katrina, is there anything that you wanted us to kind of come over? Not come over. Is there anything that you wanted us to do? All right, I'll go on. Hi, Jen. You've actually been participating in the discussion board a fair amount. Do you have any other particular questions that you have? Am I locked up? Andre's locked up.